Alright, hey guys. So for this week, you guys in lecture spoke about biosynthetic pathways. You guys also spoke about DNA and RNA and the different processes they go through. And you guys will also learn about chromosomes. So what the first thing we're going to do in this recitation is we're going to cover biosynthetic pathways. Alright, so what is a biosynthetic pathway? Also known and referred to as a metabolic pathway. So essentially, a biosynthetic pathway is a series of enzyme-mediated reactions in which one reaction is used to facilitate the next by acting as a substrate. And so an example of this would be the citric acid cycle. So as you can see right here in this image, we have an X-ray and we have Conidia, which is a type of, it's an organism that can grow on different types of media. So the way biosynthetic pathways work are an organism is going to be introduced, or it's going to be allowed to be set in a specific medium, and it can grow. And it converts one form of the media through a series of steps into other medias or metabolites that eventually allow it to grow. So here we have X-rays or UV radiation that was used to mutagenize or to damage the DNA of this organism, and it was placed in two different types of media: complete medium and minimal media. So complete medium is a medium that contains all the nutrients essential for growth. And minimal medium, as the name suggests, is a medium that contains the minimum amount of nutrients for growth. Minimal media is generally composed of glucose and will have some inorganic salts. Whereas complete medium would contain all essential amino acids and the like. Now typically, the organism would be able to grow on both the mediums if the DNA was undamaged. The entire purpose of mutagenizing the organisms is to see if, one, the organism will grow on complete medium and not minimal medium, or if it'll grow on both mediums. So if it grows on both mediums, then the mutagenizing, I don't really know how to say the word, the process to damage the DNA was unsuccessful. Perhaps it damaged another portion of DNA, but it's not showing any damage in the biochemical pathways that we're looking for. So it's not important. In the world, if growth occurs on complete medium but not minimal media, then what that means is that you successfully damaged the pathway. And there's something wrong in the series of steps that it goes to from converting glucose into a different metabolite. And that's essentially what we're looking for. We're looking for that gene that's been defected. So if a mutant requires an additional nutrient that the unmutagenized mutant doesn't, then that would be called an attritional mutant. And that's what we can use to find out what's wrong with the pathway. So here, in this example, we can see that in complete medium, when the organism is placed in complete medium and middle media plus vitamins, minimal media plus purines and pyrimidines, and minimal media plus amino acids, growth only occurs in complete medium and also minimal medium plus amino acids. So this tells us a few things. First and foremost, it tells us that minimal media plus vitamins, and minimal media plus purines all lack something that allows for the growth of the organism. And so we can see that in minimal media plus amino acids, growth occurred. There was growth. So this tells us that the mutant must require amino acids in order to grow. And it's something that it cannot produce or convert from the other precursors. But we can see from right here that no growth occurred when vitamins were added or purines and pyrimidines were added. So this tells us that the organism probably already makes both of these. And so since it already makes it, the addition of it would not facilitate or allow for the growth of the organism. So what you can do with this information is, you can go and you could take from the minimal media plus the amino acids. You could take minimal media and you put different types of amino acids in different vials. You can group them up in pairs or in however many you would like, or you can just put them all, one amino acid for each vial. And the next slide, we'll see that when that does occur, and that you put all the nutritional media mutants in the vials with the different type of mediums. So we have complete medium here, minimal media, minimal plus leucine, alanine, tyrosine, and phenylalanine. We find that in complete medium and in minimal media plus tyrosine, growth does occur. So this tells us that the mutant lacks the amino acid tyrosine. Or it lacks the gene that allows for the making of tyrosine, or has a defect in it. Now, it's also important to note that leucine, alanine, and phenylalanine did not help in the growth of the mutant, which means that this mutant is unable to convert these amino acids into tyrosine. And that's a form of that's also a form, like something that occurs in the biochemical pathway. What can happen is you can have a certain amino acid, and you can use that amino acid and use enzymes to convert that amino acid into a different amino acid. But in this case, it was unable to convert any of these amino acids into tyrosine. So, by growing multiple mutants on medias supplied with different precursor molecules, mutations can then be ordered in the biochemical pathway. Alright, so here, let's set up an example. Here we have molecule X. And that's going to molecule Y, which then goes on to molecule Z. Molecule X is converted into molecule Y by enzyme A. Molecule Y is converted to molecule Z by enzyme B. Now this right here is the final product, and that allows for growth. So let's say if we had a mutant enzyme A and we had the molecule X. Would growth occur? The answer to that would be no. You would not have any growth occurring because molecule X is converted by enzyme A into molecule Y, which is then converted by enzyme B into molecule Z, and this ultimately allows for the growth. Now, if we had, for example, mutant enzyme A with the addition of molecule Y, would that allow for growth? Yes, that would allow for growth because the enzyme is only impacted in conversion of molecule X to molecule Y. It has no effect on molecule Y to molecule Z, or from enzyme or on enzyme B. So if you were to add anything that comes after the mutation or the defect in the enzyme, or the defect in the gene, such as a type of medium or a precursor molecule, which is another one, then growth would occur because that mutation has no impact on the molecules or any of the or anything up to this side, to the right of it. Now here in this example, we have mutant media right here, and we have enzyme encoding genes right here. So these right here are all of the intermediates. These are intermediates in the pathway. All right. So here are all the genes that are mutated, and the way that they're kind of named or numbered doesn't really have a specific format. You kind of just, as you go down and you find the gene, you just kind of call it, you know, this is, for example, the mutation in this gene, oh, tyrosine A, we're just going to label it A, B, second, third is C, D is fourth, E is fifth. The namings don't really matter. Now, in minimal media, you have no growth at any of the different genes, right? The mutations. When you add tyrosine, you have growth at every single mutation. When you add HA, you have growth at tyrosine A and tyrosine D. When you add MA, you have growth at tyrosine D. And when you add 4-HPP, you have growth in tyrosine A, tyrosine B, tyrosine A, tyrosine C, and tyrosine D. When you add 4-HPP. Now, the way, or what you're supposed to do with this is you're supposed to try to organize this into a biochemical pathway, as we did up here with molecule X, Y, and Z, the enzymes. So what you would do is you would order the intermediates from minuses to pluses, left to right, from lowest growth to most growth. So you would go around and you'd read, from minimal media, it had the least amount of growth, tyrosine had the most amount of growth, and we'll put this all the way on the right side, and we'll put this right here all the way on the left side. Now, if we look in the pathway, we see that HA has growth in two areas, or in two different genes, mutations. MA only has growth in one. HPP has growth in three. So this would come right before tyrosine. HA would come right before HPP. MA would be right after minimal media because it had the least amount of growth in, with regard to these other intermediates. So if we were to do that, we would find that we have minimal media, MA, HA, HPP, and tyrosine, with tyrosine being the final product because it revives or recovers all the mutants. And to recover a mutant is essentially to allow growth for. So what this means is we have minimal media, also known as a precursor. So essentially what a precursor is, it's a, to define it, it's a compound or a molecule that participates in chemical reactions that produce another compound, in a chemical reaction or multiple chemical reactions. So here we have the precursor molecule, which is then converted into Ma, which is then converted into Ha, which is then converted into 4-HPP, which is then converted into tyrosine. Now, reading this, we see that from minimal media to Ma, growth occurred in tyrosine D, the gene that's labeled tyrosine D, which would mean that tyrosine D is the missing link, or the enzyme that codes from going to the precursor or from minimal media to Ma. So tyrosine D converts the precursor or minimal media into Ma. If we look at HA, which is the next step of the pathway from how we organized it, we find that tyrosine A now has growth, which would mean that tyrosine A is the gene that converts Ma to HA. Going on to HPP, we find that tyrosine C has growth, in which it didn't have growth before, which would mean that the conversion from HA to HPP is facilitated by tyrosine C. And now, we see that from HPP to tyrosine, there was growth in tyrosine E and tyrosine B. So what that means is that both of these allow for the conversion of HPP to tyrosine. Sorry guys, the video cut out, but this would mean that there are two genes which facilitate the conversion of HPP into tyrosine. So we would have minimal media, which goes to Ma, which goes to HA, which goes to 4-HPP, which then has two separate pathways which allow for the reduction of tyrosine. All right, now here's an example that I would like for you guys to pause and to try to work on. All right, now let's get started on solving it. So here we can see that there are mutations in Phi1, Phi2, and Phi3, and we have minimal media, which is the precursor, pyrimidine, thiazole, and thymine. Now, fortunately for me, or for you, these are already organized. We see that in minimal media there was no growth. From minimal media to pyrimidine, there was growth at Phi2. We see that from minimal media to thiazole, there was growth at Phi1. And we see that thymine has the most growth. Now this is a bit confusing because from pyrimidine to thiazole, there is no growth at Phi2. So if this followed the format that pyrimidine goes to thiazole, then you would also have to have growth at Phi2 since it grew at pyrimidine, and this would come before it in the pathway. But in this case, it doesn't. And what that means is you have two separate pathways to produce thymine. So you have minimal media, all right, so you have minimal media, which then goes to pyrimidine. But it can also go to thiazole, which then both of these go and produce thymine. So here, from minimal media to pyrimidine, we have Phi2. From minimal media to thiazole, we have the use of Phi1, a mutant. This enzyme produces thiazole. And now to go from either one of these, Phi3 is used to convert both of these into thiamine. So this right here, if we were to add this into the mix, this would rescue all of this because it would allow for growth at every single mutant because it comes in the end of the pathway. And a way to think of it is if we have a road. Here we have a road, and there were some motorcyclists, and they got in a car crash right here. Now, the road's blocked off. No one can cut. No one can go. No one can move here. They're stopped right here by this block. Now let's say that we had a road that converges after the block. So this car right here wants to turn in this one-way road. It's able to continue going. It's not the best car, but I hope you get the point. So anything that comes after the roadblock is not affected by the roadblock. So anything that comes after the roadblock cannot be impacted by the roadblock, if that makes sense. All right, now let's talk about mutations. So this is what we spoke about this last week, and there's different types of mutations. There are 3 types of mutations: base substitutions, deletions, and insertions. And so we have point mutations, and point mutations are mutations in which one nucleotide base is changed. And within point mutations, you have a variety of different types. You have synonymous mutations, and synonymous mutations are mutations that change a single nucleotide that does not result in a change in the amino acid sequence, or that's coded for. So in this example right here, we can see that from the original DNA corresponds to leucine. If we were to change the U right here to a C, it still produces leucine. So this would be a synonymous change. It would not impact the protein that's produced. It would not impact the amino acid sequences that are being coded for. Now, there are also non-synonymous mutations, which are changes in a single nucleotide that result in the change of the amino acid. So here, we see that in the other example, if it was changed to a G, that would produce a different amino acid in total. So that would be a non-synonymous mutation. Now there are also deletions and insertions. So deletion is a mutation in which a nucleotide base is deleted. So this right here would be entirely removed. And that would shift all the other DNA sequences after it. So if we were to have, for example, UAA, AGC as a nucleotide, as a DNA sequence or nucleotide sequence, or this would be RNA since it has a U. But if we were to have this this sequence, and this right here, this U right here was deleted. Oof. We would end up with UA, since this right here was deleted. So that would shift all of this to the side. So what we'd end up with is UAA, then we'd have GC, and it'd have the other nucleotide from the molecule or from the nucleotide sequences that come after it. That's also the same case for insertions. If we were to insert an extra nucleotide, it'd be similar in the sense that all this would be shifted to the other side. Everything after the shift would be shifted. And now there are also nonsense mutations. Now nonsense mutations are mutations in which a stop codon is randomly inserted. So we would have, for example, just a beginning sequence UAG, and then it'd code for example, an amino acid, GUA, and then all of a sudden it would just code for us, it would just say stop. So we would have start, val, and stop. It doesn't make any sense. It's nonsense. And so that's in the name. On to the important parts of DNA and RNA sequences. Here, let's say we have an entire sequence. We have the 5' untranslated region, exons and introns, '3' untranslated region, and here we have TSS, which means transcriptional start site. And here we would have another TSS, but this would be transcriptional stop site. So let's say we had DNA. We have transcription factor binding sequences. We have a promoter region. We have the 5' UTR, an exon, we have an intron, we have another exon, we have a stop codon, and we have a 3' untranslated region with the transcriptional stop site. Pre-RNA, what we would end up with is the 5' untranslated region, exon, intron, another exon, and the 3' untranslated region. Now, RNA goes through a maturing process in which certain steps occur to make the final mRNA that will be translated into amino acids and proteins. So to go from pre-RNA to mature mRNA, what happens is a 5' cap is added, and then we have the 5' untranslated region. We have introns, which are included, and the introns are the coding sequences. We have the 3' untranslated region, and we have the addition of a poly A tail. So what happens is we have introns that are included, or pardon me, I'm incorrect in this. No, we have introns that are spliced out. Introns are spliced out. And exons are included in the mature mRNA process. Now it can get confusing because I like to think of introns as included, and it can kind of, I guess, confuse you sometimes. So just think about exons as, there's a way to think about it that'll make it, I guess, stick in your head a bit better. Think of exons as, I suppose, extra important. Okay, so think about exons as exons encode for introns are insignificant. So exons encode, introns insignificant. So, and exons are the only sequences that get translated. So the 5' cap, which is added, the function of that is to protect the mRNA from degradation. And it also assists the ribosome with translation. The poly A tail which is added it also protects the ribosome from degradation, don't know how to say that word. It also aids in transcription termination and mRNA stability. And we also have introns. So introns do not carry information to produce desired protein. It has what we could refer to as junk information, and so they're spliced out and removed. Now onto the untranslated regions, UTRs. The 5' UTR, it's directly upstream from the start codon. It affects the binding of polymerase and transcription. It also has a sequence that allows the ribosome to bind and initiate. Now the 3' untranslated region is directly downstream from the stop codon. and that plays a role in transcription termination and post-transcription modification. It also aids in mRNA stability. All right, now going on to differences between homologous pairs and sister chromatids. All right, so here, I'm gonna draw a little diagram of two chromosomes. All right, so these are both chromosome 1. Now, a homologous pair is a chromosome that shares the same structural features and has essentially the same gene sequences, but they made different alleles. So, this chromosome and this chromosome right here, they have the same structure, same location for genes, but they differ in the allele sequences, or they may differ. So one of them is maternal and the other is paternal. Let's say this one right here is the maternal, this one right here is the paternal. And for example, this portion of the gene, this may code for eye color. Let's say that the father, he codes for, he has an allele for brown eyes, whereas the mother has an allele for blue eyes. Now they're still in the same location and they code for the same gene for the same protein. They just have the same type of protein, they just differ in the sequence that produces the protein. So they code for different eyes, but they are essentially the same chromosome, just minor differences in alleles. Now, sister chromatids what they are is they're identical replicates or copies of the same chromosome. So let's take this chromosome right here, the maternal chromosome. Here we have a centromere and we have duplicates of each side. So this side right here is an exactly and replicated copy of this side right here. Normally, these chromosomes, when next to each other, would look kind of like little lines. Have like chromosome 1, chromosome 2. During meiosis, in which the DNA and the overall chromosome is replicated, what you get is you get sister chromatids. So sister chromatids are just replicates of these portions of this chromosome right here. So these centromeres right here, they would dictate whether it's considered one chromosome or more than one chromosome. So this is still one chromosome, but it has a replicated copy, a replicated sister chromatid. So when replication is occurring, what's going to happen is, let's say that these are the centromeres right here. This side is going to copy and become similar to the other side. It's going to be an exact identical copy to the other to the chromosome. So sister chromatids are just identical replicates of a chromosome. Now this process also occurs in mitosis, but in meiosis what happens is you have homologous pairs and they line up next to each other. So here we have both chromosomes before replication begins. They're next to each other. Here we have the centromeres. Now the chromosome is beginning to replicate and it's going through the process of interphase. And so what you have are the sister chromatids just now appear. Then you have meiosis 1, and within meiosis 1 you have the same steps as mitosis, which have interphase, and then you go through the process of prophase, and metaphase, anaphase, telophase, and you have cytokinesis. So what happens is, they both the homologous pairs, they line up next to each other, and they're split. One chromosome goes to one side, the other chromosome goes to the other side. So here, let's label this one right here as paternal, maternal, paternal, maternal. So we have the paternal chromosome that's replicated on the left side, and we have the maternal chromosome that's replicated on the right side. That's what occurs in meiosis 1. In meiosis 2, what then happens is you have the sister chromatids, which then separate from one another. So this side of the central chromatid is going to go into one cell, whereas this side is going to go into another cell. Likewise, it happens on the same to the other chromosome. Now, if you were to, for example, be given a problem that's asking you, let's say we have two genes right here. We have chromosome 1, which is replicated. Two replicated homologous pairs, and let's say this codes for a big R. This right here codes for a little r. That's not a little r. Now, since this is an identical replicate of the other side, this right here, the opposing side will have a little r as well. This opposing side will have a big R as well. So when they go to split, during the first meiosis, you're going to get the homologous chromosomes which separate. And that's going to give you big R, little r. Now these are going to split once more. During meiosis 2, the sister chromatids will split. It'll end up with chromosomes that have one of the alleles. Big R, little r, little r. Now, if you were given a problem, let's say, for example, you have a chromosome right here. You have a cell right here, and it has two types of chromosomes, two pairs. You have homologous pair 1 and you have homologous pair number 2. And this right here codes for, let's say, gene B. This one codes for a big B, this codes for a little b. This right here codes for gene A, you have a big A, and here you have a little a. Now, if you were asked a question that says, give me the probability, what's the probability of having a chromosome, or of having a cell that contains both big A, little b. Now, what you would do is, or an easy way to do it would be to separate it, and you have, here you have big A, little a, little a. You would then go through all the processes, and you would end up with a, little a, little a. So you would find that half of the cells, or half of the gametes produced, would be big A or little a. Now, likewise, on the other side, you have big B, little b, little b. And I'd go through the processes of meiosis again, and you would end up with 4 haploid cells, which would contain big B, little b, and you have the same probability. Half to get big B or little b. Now, the question is asking, what's the probability of getting a cell that ends up with both of these? Now, assuming that crossing over and all this other stuff doesn't occur, through independent assortment, what you would just do is you would take both the probabilities, which would be half. You'd get half, or 1/2 chance probability to have big A, and you multiply that by the other half probability to get little b. So 1/2 times 1/2 is 1/4, which would be 0.25. So that is the percentage probability of ending up with a gamete that contains this genotype.